All right, let's get started for this afternoon. Frances Champagne is an associate professor in the Department of Psychology at Columbia University. Dr. Champagne's research has focused on the neurobiology of maternal care and the epigenetic effects of mother-infant interactions. Specifically, Dr. Champagne has been a leader in the field of epigenetics, where her research with rodents has demonstrated that the quality of maternal care received in infancy can lead to long-term changes in offspring gene expression and behavior. Dr. Champagne's current and ongoing research explores the implications of these influences for the transmission of behavior across generations and the molecular mechanisms through which these effects are achieved. Among her other achievements, Dr. Champagne is the 2007 recipient of the NIH Director's New Innovator Award. Please join me in welcoming Dr. Frances Champagne.
Thank you for that lovely introduction, and it's a pleasure to be here, and thank you, Darsha, for the invitation. It's a very nice, broad meeting where we have a lot of different aspects of parental behavior and development. So most of my research focuses on how prenatal and postnatal mother-infant interactions can shape development, and certainly there's evidence for this across a wide variety of species. In humans and primates, and certainly in the rodents that we study in my lab. And certainly the brain during prenatal and postnatal development is really open to those kind of experiences. We're in an active phase of development. There's massive changes in the arrangement of neurons within the brain. And so this is a window during which experiences can exert a long-term effect. The question that the phenomenon of these effects raises is that of mechanism. So how is it that variation in the experiences that individuals have during both the prenatal and postnatal period lead to long-term developmental changes? And so I'll show you a little bit about how we've been tackling that mechanistic question. The second question is, can these developmental effects persist across generations? And this is a very growing and exciting field of study where we look not just at mechanism, but at the heritability of these developmental changes. And finally, hopefully I'll have time to discuss this in a little bit of depth, is what would be the adaptive significance of having a system that whereby these within and across generational effects occur. So hopefully we'll tackle the issues of mechanism, inheritance, and adaptiveness today. Now, when we're thinking about mechanism, there's certainly no shortage of options to choose from. And certainly mechanism can be considered at a number of levels, at the behavioral level, at the neurobiological level, but here I'm going to talk about the molecular level and how really experiences come to be embedded within our biology. Now, if we look at the literature on early life experiences and their long-term effects, certainly we see altered neurodevelopment. So we're trying to understand the origins of behavioral variation, and so neurodevelopment is going to be central to that. And of course, one reason for that altered neurodevelopment is long-term changes in gene expression, in the activity of various genes within various neural circuits within the brain. And what we invariably see is that early life experiences have effects on gene activity that persist long beyond the actual experience itself. So if we think of prenatal events, this is occurring during a very brief window relative to the lifespan of an individual, and yet those changes can persist. Those changes in gene activity can persist well into adulthood. Now, I'm going to explore this issue of gene expression changes from both the prenatal and postnatal perspective. And first I'll start with some of the work we've been doing on prenatal stress and how the stress that mothers experience can come to shape the fetal brain. And certainly within the human and animal literatures, there's significant evidence for these effects. The brain and body are rapidly developing during this period, creating a window of opportunity during which experiences can come to have a long-term effect. Now, of course, one of the major prenatal events that can have a significant impact for long-term neurodevelopment is maternal stress. And this is referred really as the programming of the stress response through fetal exposure to maternal stress. And so mothers experience stress, and that comes to affect fetal development in some way. And consequent to that effect, we have a risk of prematurity, lower birth weight, increased stress response, and increased risk of depression and anxiety in humans. And the question again is that of mechanism. Now, if we look at the dynamics between mothers and fetuses during this prenatal period, we have to consider the placenta. The placenta is the major interface between these two organisms, and anything that can perturb placental function can lead to long-term developmental changes, both in growth and metabolism, but also in long-term neurobiological outcomes. Now, mothers, when they experience stress, psychological stress, exhibit increases in glucocorticoids. So that's cortisol in humans and corticosterone in the rodents that I'm going to talk about. Now, we have built-in mechanisms to protect us from these elevated glucocorticoids, and the fetus certainly has a built-in mechanism within the placenta. The gene 11-beta hydroxysteroid dehydrogenase 2, 11-beta HSD-2, is an enzyme that inactivates active glucocorticoids. So it basically takes those stress hormones that we know to have such a profound effect on our physiology and behavior and inactivates them, puts them into a form where it can no longer activate the receptors for these glucocorticoids. Now, the placenta can metabolize about 80% of maternal glucocorticoids, okay? So 80% of the circulating maternal glucocorticoids do not pass to the fetal compartment. And this is a nice protective mechanism. And this gene, 11-beta HSD-2, is essential for this function. Now, we've been exploring how stress affects the activity of this particular gene. So we're interested in gene expression as a mechanism through which early life experiences exert long-term changes. We want to focus on this particular gene. Now, if we look at the expression level, so how much of this gene is expressed within the placenta, fetal cortex, and hypothalamus, what we find is what stress does, what maternal stress does during gestation, is to decrease the levels of this enzyme. So what does that mean? Well, you've got less 11-beta HSD-2. That means more glucocorticoids can pass across the placenta and affect the fetal brain. Now, having some glucocorticoids pass into the fetal compartment is extremely important for lung development, okay? So what glucocorticoids do is enhance lung development. However, if you are inundated with very high levels of glucocorticoids, that can have a significant impact on the stress physiology of the developing fetus in the organization of the circuits that will be responsible for HPA function in later life. And so what maternal stress is doing is allowing more of those glucocorticoids to cross and affect these systems. Now, there could be an adaptive rationale for this. If you're a pregnant female and under a severe amount of stress, accelerating lung development in anticipation of preterm labor might be an advantageous adaptation. So, we can think of why this would occur, but the end result is that the cost of having these well-developed lungs, where you can exist without the mother's circulatory system, is a stressed brain. Now, a question raised by this finding and many other findings looking at gene expression differences in various tissues as a function of a variety of life experiences is that of mechanism. So, what mechanism could allow for the sustained change in gene activity within these tissues? And it is this question that led us to explore epigenetic modifications. Epigenetic modifications are molecular modifications that affect DNA expression, gene expression, without affecting the underlying sequence. So what you can think of is the DNA sequence provides one level of information, biological information. Epigenetic regulation of that DNA sequence provides another layer of information and can ultimately serve as an on-off switch. So what I'll do is walk you through the basics of one of the key epigenetic mechanisms that we study in my lab, that of DNA methylation. So here's a little cartoon illustrating kind of the structural aspects at a molecular level that we've been looking at. So here we have DNA. And DNA in our cells is wrapped around clusters of proteins. Okay? For the most part, our genes aren't expressed. We don't want to be expressing genes all the time. We want to be expressing genes at the right time for a needed function. And our DNA is wrapped around this core of proteins called histones. And this wrapping prevents gene expression. Okay? So if you've got a lot of proteins around a sequence of DNA, it can't really do much. It can't be expressed and create any kind of product. Now, in order for expression to occur, in order for genes to become active, they need to come unwrapped from all of these proteins, which they are condensed in the cell nucleus. And so RNA polymerase, which is the main enzyme that comes and activates DNA and creates a product, a gene expression product, needs to bind to DNA. And that DNA needs to come unwound from the protein. So really, accessibility of DNA is the key issue in epigenetics, making DNA more or less accessible. Now, one modification to DNA, an epigenetic modification, which affects this accessibility is DNA methylation. Now, DNA methylation is a chemical modification to DNA whereby a methyl group, a chemical, that we have naturally. We ingest foods that contain these methyl groups, and so it's a natural biological process. These methyl groups become chemically added to cytosines within the DNA sequence. So now instead of a cytosine in the DNA, we have a 5-methylcytosine because it's got this little additional methyl group on it. And this chemical reaction involves DNA methyltransferase. So this is an enzyme that does all the heavy lifting in terms of this particular modification. Now, if you're DNA and you have methyl groups all clustered around you, it's very hard to access the DNA sequence. So this is one way to shut down gene activity. Now, DNA methylation is typically a suppressive epigenetic modification, which means it does turn off gene activity. And it also is potentially heritable. And so, as I mentioned, I'm interested in both mechanism and heritability. And so this makes it a very enticing mechanism to study. So I'll show you what I mean by heritable. So here we've got a little schematic of a cell. And in green, we have a little squiggle of DNA. And within that DNA, we have 2 methyl groups in this particular illustrative example. Okay, so we've got cell, we've got the DNA, and we've got methylated regions of DNA. Now, when cells divide, the biological logic of cell division is that they pass down, they copy their DNA, and they pass it down to their daughter cells, which they certainly do. But they also pass down these DNA methylation patterns. They're also heritable. Okay, so you must reliably copy your DNA, and you must reliably copy your DNA methylation. This is an extremely important phenomenon to occur because cells can't simply revert to some earlier ancestral form. They need to maintain their phenotype. If all your neurons started dividing and becoming stem cells, it would be a little bit problematic. Cells pass on their characteristic through DNA methylation. Now, we can add a new methyl group, so we've taken a little methyl group and added it to this DNA. This is a process called de novo methylation, where we've added something new to the DNA. Importantly, that will change the phenotype of the cell. Okay, so DNA methylation affects gene activity affects the characteristics, the functioning of the cell, and now we've changed that cell. And importantly, when those cells divide, they will pass on both their DNA and their DNA methylation pattern. This is the mechanism through which cellular differentiation occurs, okay? So the reason why all the cells in your body, for the most part, have identical DNA, but are functionally very different. Your brain cells are different from your skin cells, they're different from your muscle cells. So we have a range of phenotypes, characters in our cells, which are encoded through these epigenetic mechanisms. Okay, so this is very molecular and what is going on as a normal process of biology. Can we take that to gain a better understanding of how the experiences of the individual come to be embedded at a biological level and shape their neurodevelopment? And so can epigenetic modifications and epigenetic variation lead to long-term changes in gene expression and have all these consequences. Now, historically, it was thought that once you are beyond the very early stages of embryonic development, there was no possibility to change your epigenetic patterns, especially DNA methylation. So it was thought that everything was in stone at an embryonic time point, and these were not particularly plastic types of mechanisms that we should be looking at when we're considering prenatal and postnatal events. But I think one of the great gains in the recent literature is a greater understanding of how plastic and malleable these epigenetic factors are, and it seems that part of learning and memory involves epigenetic changes. So even in the adult brain, as you're learning something new and consolidating that into memory, you require the process of DNA methylation. It's a way that events become embedded at a biological level. Okay, so can we use this knowledge, this advances in molecular biology, to better understand how the experiences of an individual during prenatal and postnatal development come to be embedded? Here we're looking at DNA methylation within the 11-beta HSD-2 gene in the placenta. So we know that stress causes a decrease in the level of this enzyme. If we look at the DNA methylation pattern within this gene, so on this axis here we have different sites within the DNA sequence of the gene, and this is the percent of methylation at this site. And if we can compare control and stress females and look at the placenta, what we find is that several regions within that 11-beta HSD-2 gene, we have increased levels of DNA methylation. So increased levels of DNA methylation tend to shut genes off, and in this case, it's shutting off a gene that is really critical for buffering the effects of stress on the fetus. So we've shut off this gene epigenetically, and as a consequence, we're getting a highly stress-sensitive brain. Now, I mentioned the DNA methyltransferases, those enzymes which are quite critical for the DNA methylation process. And they're not just critical for a very abstract molecular event, they're critical for survival, okay? Now, if we were to mutate the genes for the various DNA methyltransferases, so there's three typical ones that are studied currently, so DNMT1, 3A, and 3B. These genes are necessary for development. You will not survive if you don't have these genes. So in mouse models, it's shown that if you delete the gene for these DNA methyltransferases, it results in lethality. So the pups die. They die a little later in life if you mutate the 3A than the 1. DNMT1 is responsible for that process that I showed you during mitosis. When cells divide, they have to copy that DNA methylation pattern. If you lack DNMT1, you can't do the copying, okay? So that's a copying enzyme. The DNMT3A is adding new methyl groups to the DNA. And you can survive a little longer with it, but postnatal, you still don't survive. So these are incredibly important in considering the epigenetic process of developmental experiences. And perhaps not surprisingly, what we see in the placenta of stressed dams is that they're having increases in DNMT3A. They have more DNA methyltransferase, more DNA methylation, less activity of genes. And we're only looking at one gene here, but the effect on this enzyme, which is not gene-specific, could mean that a number of genes within the placenta and perhaps even the fetal brain can be affected. Okay, so we have a process here whereby the stress experienced by mothers during pregnancy affects epigenetic pathways within the placenta, leading to altered gene expression and the brain, leading to a heightened stress responsivity that can be observed even into adulthood. Now, an important alternative pathway to this direct hormonal, perhaps hormonally driven pathway through the mother's stress is the way stress experienced during pregnancy affects maternal behavior. And certainly it does. Very high levels of stress can lead to a significant downregulation in maternal behavior, decrease various aspects of mother-infant interactions. Acute stress can actually stimulate maternal behavior. So we have to think about the chronicity and level of stress when we're considering these effects. And indeed, what we find is that in this model, gestational stress induces persistent decreases in maternal care, and in some cases is the key pathway shaping stress response of offspring. Now I'll talk about another prenatal exposure to which we were all being exposed, regardless of our stress levels, and that's the various toxins within our environment. And so, working with the Columbia Center for Children's Environmental Health, we've been looking at a number of pollutants, some that are particularly high in the Manhattan area, others which are high everywhere. And the one that is high everywhere is bisphenol A. Bisphenol A is an endocrine disrupting compound, so that means it disrupts various endocrine pathways, and it's used in the production of plastics. Okay, so it's used to enhance plastic production. However, it can leach out of the plastics when they're warmed up into our food, into our water. And there is growing concern that this could have a long-term neurobiological, metabolic, and immune consequence for the population. Now, currently, if you go into a baby store, everything will say BPA free. And this is because of the concern over bisphenol A. However, they seem to be concerned about babies and children, but not so much the rest of us. So we're trying to work towards looking at what level of BPA is safe or unsafe, as you'll see. Okay, so we've been using mice in the lab, and we've been giving them different low doses of BPA. Now, just to put these doses in perspective, so the doses are down here, so we've got non-exposed, and they're not really non-exposed. Trying to get rid of BPA even in a laboratory environment is almost impossible because everything is made of plastic, but there's various things we can do to limit exposure. We have 2 microgram, 20 microgram, and 200 microgram per kilogram of weight of BPA a day. Now, currently, the EPA considered safe exposure level is right about here, okay? So we're using doses that are below and above what's considered safe by the various governing bodies. So this is looking at gene activity. And now we're not looking at 11-beta HSD-2, although interestingly, it is also decreased as a function of bisphenol A exposure. We're looking in the brain, in the hypothalamus, of individuals who've had prenatal exposure to this compound. And what we see is that if we look at the activity of estrogen receptor alpha and estrogen receptor beta, these are 2 estrogen receptors. We think that BPA is particularly targeting the estrogen system, so these are important targets to think of. We see some very bizarre effects of BPA that are dependent both on the sex of the individual and the dose of BPA that they're being exposed to. Now, in females, we see a U-shaped response in terms of the activity of this gene in response to various, in response to dose. And so vehicle treated have, a certain level of estrogen receptor alpha that's suppressed with very low dose of BPA and then starts to normalize when we get into the higher levels. These kind of U and inverted U functions are very typical of hormonal responses. And you can think of stress as a good example of this, where a little bit of stress goes a long way, a lot of stress can actually halt development quite significantly. And of course, the pattern that we see with females is quite different from the pattern we see with males, where they show a slight increase in expression and then a significant decrease as we getting into the higher levels. And the same pattern is seen at when we look at the activity of estrogen receptor beta. So clearly, prenatal, an isolated prenatal exposure to this compound can have persistent effects on gene activity within the brain. Now, we can also look at the levels of those DNA methyltransferases. Okay, so DNMT, we're looking here at DNMT1 and DNMT3A. And what we find is that, again, we have the females and males exhibiting very different responses in terms of the activity of this gene. So if we, at very low dose exposures to BPA, we have less of these DNA methyltransferases, and then we start normalizing when we get into the high levels. Now, importantly, as was the case for prenatal stress, these genes don't have specific target genes. They can target the entire genome, which means there could be widespread epigenetic effects within the genome as a function of very low doses of exposure to these toxins. And we've been looking at DNA methylation within those estrogen receptors. And again, as you might predict from the expression levels, we see sex-specific, brain region-specific, and dose-specific effects. But certainly it's affecting DNA methylation, which can mean there can be very sustained effects. Now, an interesting phenomenon with prenatal bisphenol A exposure is its effect on postnatal maternal care. So we're seeing similar parallels to the study on prenatal stress. And so if we look at two forms of rodent maternal care, one being pup licking grooming. So during the postnatal period, there's quite a lot of licking, grooming of the pups. This serves a number of physiological functions. And in fact, rodent pups can't urinate or defecate unless they have a minimal level of tactile stimulation, either from the mothers, or if you're rearing them artificially, you can give it with a paintbrush. And of course, frequency of nursing. Both of these show these same curvilinear effects, showing decreases with the low dose and increasing with the high dose of bisphenol A. Now, given this pattern of postnatal maternal care that's induced by bisphenol A, you might ask, well, is it the modification of postnatal maternal mother-infant interactions that is shaping all of these gene expression effects in the brain? And what's interesting is we find that no, in fact, maternal care has its own effect. And bisphenol A has its own effect. One is the maternal care is actually not mediating effects of the BPA. They're both contributing to your overall level of activity of these genes. Now, what's interesting about the two examples that I've run through is that although they are done in rodent studies, they are paired with human studies. So one of the questions that you might have is, well, how can we apply the study of these epigenetic mechanisms in human populations? And that's what's one of the great advances with some of the methodologies developed in this field has allowed us to do. And so in the context of prenatal stress, we're working with mothers who experience depression during pregnancy and looking if we can find some epigenetic modification which will predict long-term changes in offspring, reactivity to novelty, and stress response. Now, obviously, we can't look at the brain in humans, but we can certainly look at the placenta. So we've been collecting placentas for the last 2 years, and we can look at gene activity and DNA methylation within those placentas. In the case of bisphenol A, we are likewise collecting blood samples from human mothers who are exposed to varying levels of BPA, and we're going to see whether that exposure predicts various outcomes in their children. So we're looking at aggression, peer relationships, and various growth and metabolism markers. In fact, obesity has been linked to exposure to these endocrine disrupting chemicals, and so it may be one of the pathways through which the increase in obesity is being achieved. Now that brings us to the postnatal period, which I've already highlighted through its interaction with the prenatal period. Certainly it's very hard to tease apart the two. Any exposure that occurs during prenatal or gestation can have a long-term effect on the postnatal period. And of course, when we think about postnatal maternal care, we have lots of examples from the human and animal literature that variation in postnatal care, you don't even have to induce it, but variation in care can induce a variety of outcomes and variation in brain and behavior. Now, these are the mothers that we've been studying in my lab for quite some time now. This is a Long Evans rat, and this is her litter of pups. Now, our rats have between 16 and 20 pups at a time, and they only have 8 nipples, so they have to kind of do quite a lot of activity and maneuvering around the litter. Now, you might not be able to see this, but this female is licking this pup. So the pups are sitting under, and she's giving it a good old lick, helping it to urinate and defecate. Here we see licking and grooming again. This is a very tactile aspect of maternal care, and certainly some of the immediate physiological responses of pups are very similar to what you would see if you took a preterm infant or a newborn and provided it with tactile stimulation. So if it's hard to relate to the actual licking itself, we can think of this as a critical form of touch that conveys a variety of long-term effects. Now, one note about the video you just saw. So this was a rat with its one litter. This is how laboratory rodents throughout laboratories around the world are reared. But in fact, in light of some of the discussions we had this morning, it's important to note that this is not how rodents rear their offspring in the wild. They rear communally. And if you allow rodents to rear their offspring communally, where they have a number of lactating females pooling their pups into a big pile, you see a number of very interesting effects. Communal rearing increases the level of maternal care that any individual female exhibits towards the litter. And females will care for non-kin. In fact, in our laboratory studies, we can give cross-fostered offspring to which they are not biologically related at all. And they will provide very rich maternal care. And this has a number of long-term effects, very similar to the model that I'm going to show you now. Now, if we look in a laboratory where we have all of these rats that have very limited genetic variability, what's interesting is that we find that there's quite a lot of variability in maternal care. And so we can observe this is actually a cohort of about 400 rat dams and their pups. And there's a nice normal distribution in maternal care. And so what we can do is we can take those females who provide either low levels or high levels of maternal care and compare the mothers and the offspring to determine the impact of this variation on life history. Now, I'll also point out that high versus low licking grooming mothers are very interesting in terms of their preference for certain stimuli. So high licking grooming mothers, if you give them a choice between a toy and their pups, they will always choose their pups. The low licking grooming mothers, if given a choice between a toy and their pups, will almost always choose the toy. So there's something clearly different about the reward system in these females. We've looked over the course of the prenatal period up until the time of weaning and found some very striking differences as well. So the amount of licking and grooming a female bestows to her litter during this early postnatal phase is actually very predictive of when she will wean her offspring. So our high licking grooming females will wean their offspring much later. In the lab, we actually artificially wean. So there's a designated day to which pups are removed from their mother. This is based on nutritional independence, when they can actually eat solid food. If you just allow the pups to interact with mothers naturally, they will wean themselves much later. Our low-licking grooming mothers will actively wean their pups. So they behave very aggressively to them if they try and suckle from the mother beyond that point when they can actually digest food. And so there's a number of kind of a cascade of different effects that are induced by high and low-licking grooming. Now, consequent to this early exposure, we see a number of systems that are affected by experiencing either low or high levels of maternal care in infancy. Now, one of the reasons we started looking at maternal care is because we wanted to account for the wide variation in stress responsivity that we observed within the animal colonies amongst individuals, again, that had very limited genetic variability. And in fact, maternal care is a very strong predictor of how stress responsive an individual is. The higher the licking grooming an infant receives in infancy, the more reduced its stress response will be later on in life. Now, there's a few caveats to that, but in general, that is the phenomenon that we see. And this is linked to changes in the HPA circuits and glucocorticoid receptors within the hippocampus and glucocorticoid output from that axis. Response to reward is also altered. And it's not so much, in some cases, the offspring of low-licking grooming dams appear anhedonic, but for the most part, it's that they prefer different things as rewards. Cognition is certainly altered, and if you affect stress, you affect cognition almost immediately. And so offspring that have received low levels of care often perform more poorly on learning and memory tasks, and social behavior is also affected. Now, offspring who are reared by low-licking grooming mothers actually start engaging in play behaviors much earlier than those that are reared by a high-licking grooming mother, possibly because the ones that are being reared by a high-licking grooming mother are still suckling, whereas the low-licking groomed offspring have been weaned from their mothers and are interacting with their peers. Now, one of the outcomes that has always been of interest to me is the way in which the maternal care that you experience during that early postnatal period comes to predict adult maternal behavior in female offspring. And so if we look at the adult female offspring reared by a high or low licking grooming mother, and we actually have those mids in here as well, we find that the amount of maternal care that you receive in infancy is a very strong predictor of how much licking and grooming you will bestow to your offspring when you're adult and caring for them. So there seems to be a transmission from mother to daughter of maternal behavior. And I've been very interested in the neurobiology of this effect. And what we see is that if we look, so these are schematics of coronal slices through the brain where we're looking at various hypothalamic regions, including the medial preoptic area of the hypothalamus. In mammals, this is a key brain region for maternal behavior. And if we look at offspring who've been reared by high or low licking grooming mothers, what we see is, as you might suspect, more oxytocin receptors in the medial preoptic area amongst those who've received high levels of maternal care, more estrogen sensitivity, and greater levels of estrogen receptor alpha mRNA. And in terms of the pathways through which this is achieved, this change in the activity of this particular gene is thought to be very critical. Estrogen receptor alpha levels are an important predictor of how well or how you will respond to estrogens. Okay, so estrogen binds to these receptors and goes on to regulate a number of other genes. Many genes have what's called an estrogen response element, which means they're sensitive to estrogens and can be changed by estrogens. And in this case, maternal care is altering the levels of that kind of uber organizer of the brain. Okay. Now, if we look at the time course through which these gene expression changes are occurring that we can observe in the adult brain, we see that they start emerging very early on in development. So these three panels represent the activity of the gene expression of estrogen receptor alpha, estrogen receptor beta in the middle, and oxytocin receptors. So we're looking at gene activity. Interestingly, we don't see differences at day 0. Okay, that would argue very much that this is a postnatal effect rather than a prenatal effect. When we start getting to day 6, certainly with estrogen receptor alpha, we start seeing that the highs are showing a little bit of an increase over lows in terms of the activity of this gene. When we get to postnatal day 21, which is the time at which they're being weaned from their mothers, we see the offspring of high licking grooming dams have far more estrogen receptor alpha than those who've received low licking grooming levels. So these changes in the medial preoptic area and that part of the hypothalamus that regulates maternal behavior precede the onset of maternal behavior quite significantly. So just to give you an idea, it's about day 66 that the adult females are being mated and caring for their offspring. So these changes that we see here are starting to emerge very early on development. We see the same pattern with estrogen receptor beta. Interestingly, oxytocin receptors aren't seen to be changed developmentally, and we think that's because these changes are consequent to these early changes. So you change the estrogen receptors, and that ultimately leads to changes in the adult brain in terms of oxytocin receptors. All right. So given that we're exploring this mechanism, epigenetic mechanism, as a pathway through which these changes in gene activity are achieved, we want to know whether in the adult female offspring who received high or low levels of care, is there differences in DNA methylation in those estrogen receptors that we think are so important. And indeed, that's exactly what we see. So we see that those offspring who are being reared by a low licking grooming dam have more DNA methylation in their estrogen receptors, which is shutting down those receptors and leading to long-term reduced activity of this gene. Whereas in comparison, those offspring that are being reared by a high licking grooming dam have less methylation, more expression of the gene. If we look at the developmental time course of this effect, the developmental time course pretty much parallels what we see in terms of the gene activity. We don't see much of an effect early on, but by postnatal day 21, those offspring who are getting lots of licking and grooming have less DNA methylation, and that effect persists to when they're lactating and caring for their own downs. So there are developmental origins of these differences in maternal care and that emerge as a function of maternal care. Okay, now given that these juveniles, so day 21 is really kind of a juvenile adolescent time point for our rodents, that we see these changes in the brain. Is there a behavioral consequence of this? And indeed, we find that juvenile females who are not normally interacting with pups, not in a lab setting, certainly, are more responsive and caring towards pups if they were reared by a high licking grooming dam. So this behavioral measure is looking at latency to engage in a maternal response. Now, for most non-lactating mammals, it takes some time to develop any kind of approach behavior towards a pup. Certainly in the rodents, this is true, and they find new pups very scary at first. But in fact, what you see in our offspring that received high levels of maternal care, they get over this quite quickly and start behaving like mothers towards these pups, even though they're not lactating. And you can see this is the percentage that have attained kind of a maternal response. So we're seeing changes in epigenetic pathways early in development. We're seeing changes in gene activity, and we're seeing changes in behavioral characteristics that would likely predict the fact that these females are going to engage in more maternal care towards their own offspring. Now, recently, we've been doing genetic manipulations of the estrogen receptor. So what we can do is go into the brain at postnatal day 6 and actually genetically reduce the levels or increase the levels of estrogen receptors in the brain, and then look at how these females do when they're juveniles in terms of maternal responsivity. In fact, what happens if you increase levels of estrogen receptor in the developing brain, you can make these females better mothers as juveniles. So they'll behave more maternally towards pups. So this would argue quite strongly that estrogen receptor is probably a key junction point in terms of predicting maternal care in our offspring. All right. Now, as a consequence of these maternal effects on the developing maternal brain, we have a transmission over generations of maternal behavior. So if you look at mothers' licking grooming levels, offspring's licking grooming levels, and grand offspring licking grooming levels, they're all very highly correlated. Importantly, this only occurs under very stable environmental conditions. So this kind of transgenerational impact of maternal care is not seen if you start manipulating the environment. You need a constant environment. This is a behavioral transfer rather than a genetic one. Now, we think that epigenetics plays a critical role in these kind of behavioral transmissions across generations. So it's a way to think about how behavior interacts with our genes in order to change behavioral phenotypes. And so we have low or high levels of licking grooming affecting DNA methylation in a gene that's very important for orchestrating the maternal brain. This affects the expression of this gene, and then as those females get older and engage in maternal behavior toward their own offspring, they're affected by this early experience of licking and grooming, which affects their own offspring's development of the maternal brain. So we have a circuit that under stable environmental conditions can lead to a very reliable transmission of maternal care over generations. And certainly in humans, there's quite a lot of evidence that there is consistency in patterns of maternal care, in terms of attachment, in terms of abuse. In primates, there's also quite a lot of evidence for this, and the suggestion that it is not due to genetic inheritance, but to these behavioral transmission pathways. Now, as I mentioned, stability of the environment is really critical for these transgenerational effects. And we know that for a number of reasons. And one is that if we manipulate the environment in any way, we can disrupt the inheritance of maternal behavior from mothers to daughters and perhaps even granddaughters. So here we have a schematic of a study in which the offspring of low or high licking grooming mothers were placed in 3 different juvenile environments. One characterized by social impoverishment, one the standard laboratory environment that all of the previous studies use, and one the enriched environment. So they have both social enrichment and toys and running wheels and everything. And so if you are reared by a low licking grooming mother, but placed for about 50 days in a juvenile environment characterized by social and physical enrichment, you become a high-licking grooming mother. Conversely, if you are reared by a high-licking grooming mother but then placed in social impoverishment during this period, you become a low-licking grooming mother. So this shows that these circuits are not set in stone, but very plastic in response to the quality of the later life environment. In fact, the environment that is far more proximal to the time of reproduction and probably very informative of what the environment will be when you do reproduce. Now, importantly, these phenotypes can be transmitted to the next generation. So they don't revert back to some ancestral phenotype. They are passing on this new phenotype to the next generation. Okay, now the inheritance system that I've just shown you is what's called an experience-dependent inheritance, okay? So there's certainly a phenotype that transmits over generations, but is behaviorally mediated. So we have mothers licking pups. This is affecting this generation's maternal behavior, affecting epigenetic pathways in the brain that stabilize the effect in the subsequent generation, and affecting the next generation's maternal behavior. This is in contrast to a very new and very interesting way of passing on epigenetic information, and that's germline inheritance. So normal genetic inheritance involves the germline. We inherit our DNA from the germlines of our mother and father. There is very interesting data coming out now that the experience of an individual over their lifetime can become encoded at an epigenetic level within the germline and be transmitted over generations. And this would lead to effects that persist in subsequent generations that you do not require this additional behavioral mechanism to transmit. So this is very Lamarckian, if you're familiar with some of the historical theories on evolution, that there is an inheritance of acquired traits. Somehow this epigenetic mark that we didn't think could persist over generations is somehow becoming encoded into our DNA and being passed down to our ancestors. And most of this type of inheritance of epigenetic variation is being studied within the context of paternal effects. So, and it gets into the question, can fathers influence the development of their offspring? From a very kind of creative perspective. And certainly fathers can affect the development of their offspring through some of the same pathways that mothers do, through the social interactions that they have with them. Now, if you think across species, what you find is that exclusive paternal care or even biparental care is fairly rare across species. So are there, in these species where there isn't social interactions between fathers and offspring, are there other ways, perhaps epigenetic, through which fathers can influence the development of their offspring? This is from a paper that's coming out in Biological Psychiatry soon, just showing how these bizarre paternal inheritance patterns can occur. So this is a model, this is an experiment where they looked at social instability in mice, okay? So they're looking at the transgenerational consequence of experiencing a lot of social stress. The social instability is achieved by mixing up social groups of rodents, which causes a lot of fighting because there's dominance hierarchies, and it can be very stressful. Now, and the blue box indicates the development of an anxiety-like phenotype. The red square, the red circle, social deficits. So males and females that experience social stress can develop deficits in functioning as a consequence. Females develop both anxiety-like behavior and social deficits. Interestingly, males only develop the social deficits in this model. Now, when you use a breeding design where you're looking at stressed moms, the descendants of stressed moms versus stressed fathers, what you see is that in the next generation, fathers don't have the stress phenotype. They don't have anxiety and they don't have social deficits, but they can pass those deficits on to their offspring. Okay? And so that's what we're seeing here in this lineage. So these are stressed, the descendants of stressed fathers who are passing on these deficits in functioning to their female offspring in multiple generations. What does this suggest? Well, they've ruled out a number of postnatal factors, and it would seem to suggest that the experience of social stress in this generation has become encoded at an epigenetic level within the gametes of the male, and they're passing this on generation after generation. And so that makes it a very interesting thing to think about the origins of stress and anxiety. It may not be from your own developmental experiences, but coming from those from where you're descended. We're also thinking of these paternal effects from a unique perspective as well. And one of the conclusions we've come to is one way in which fathers can influence the development of their offspring is via maternal care. Probably a bit biased because that's what we study, but there's some interesting evidence that this is the case. So there are genes that are only expressed if you get them from your father. This is a weird class of genes. There are genes that are only expressed if you get them from your mother. This involves epigenetic pathways, which would require a whole other lecture to talk about. They're very interesting, very important for development. And paternally expressed genes have a very significant impact on mother-infant interactions. They're highly expressed in the placenta. They're highly expressed in the hypothalamus. And deletion or disruption of these genes can impair maternal behavior quite significantly. So this is interesting because now fathers are influencing daughters' maternal care through the genes that they inherit. Another way that we've recently discovered in my lab that fathers can influence the offspring's development is through changes in the mother's behavior during the postnatal period that seem to be induced at the time of mating. Okay? So what we did is we looked at males who'd experienced a lifetime of social enrichment or a lifetime of social isolation, and we mated them with females that had had kind of a normal rearing environment. And we were actually just looking at the transgenerational effects of social isolation and enrichment. What we found, in fact, was that mothers adjusted their level of maternal care depending on the male's social experience. Now, this occurs even though the mating period is very brief. So what you do in the lab is you have the males mate with the females, and then they're removed well before the pups are ever born. And so something about the quality of those males, the behavior of those males, has changed the way that mothers interact with their offspring. And in fact, the fathers had completely changed the whole hypothalamic circuits in the mother that regulate their own maternal behavior. So these are some intriguing ways in which fathers, through kind of non-traditional routes, can influence development of their subsequent generations. Now, in just a few minutes, I'm just going to sum up, but I'll just show you some quick, interesting data that talk to the adaptiveness of some of these effects. Now, and we'll start with the notion that these epigenetic effects are tissue specific. So unlike genetics, where you can take a skin cell, you can take a blood cell, and the DNA is going to be the same, epigenetic variation is very tissue specific. And so I've been talking about the medial preoptic area, where female offspring have less methylation if they've received high maternal care. The opposite is true in another region of the hypothalamus, the ventral medial hypothalamus. And interestingly, this brain region is not involved in maternal behavior, but involved in sexual behavior. And so what we find is that offspring who've received low levels of maternal care have more estrogen receptor alpha. And as a function of this, they actually have increased sexual receptivity. Offspring who've been reared by a low-looking grooming mother actually enter puberty early and can produce more offspring in their total reproductive lifespan than those offspring who've been reared by a high-looking grooming dam. This suggests, this is just an illustration of that. So this is actually sexual receptivity, which is also altered by maternal care. So those offspring who have received high levels of maternal care are less sexually receptive. They're engaging in less kind of solicitation of sexual behavior, and they're going into puberty much later. This would suggest a difference in reproductive strategy, okay? So our offspring who are receiving low levels of maternal care may invest less levels of maternal care in their own offspring, but in a sense they compensate for this by producing many offspring. Whereas our offspring who are receiving high levels of maternal care have high levels of maternal investment in fewer offspring. Now, this strategy fits very well with what we know about the kind of environments that will promote high levels of licking grooming or low levels of licking grooming. Low levels of licking grooming are promoted by stress and social isolation, whereas social enrichment produces high levels of licking and grooming. And if you think about this in kind of a broader ecological context, if your environment is characterized by high levels of stress, high levels of predation, mortality rates will be very high. And so probably the best energetic strategy is to produce lots of offspring. A lot of them will die, but not invest all your energy in the care of those offspring, providing them enough that they survive, but not more than that. Whereas in this case, high levels of licking and grooming are promoted by enriched environments, by environments that are threat-free. And so there'll be very low mortality rates, and so investing in maternal care rather than sexual behavior would be an advantage. Both of these strategies are advantageous, dependent on the context in which you look at them. A stressed environment, this strategy would be of benefit. In a non-stressed, this other strategy. Okay, so what produces epigenetic variation? Well, there's certainly a large growth in literature now looking at every kind of experience you can possibly think of. Smoking, drug use, pesticides, hormones, nutrition, so kind of the more physical factors, but also now thinking about the social environment and the environment of early life mother-infant interactions, such as abuse, neglect, stress, nurturing. All of these experiences can have effects on those same epigenetic pathways. So whether it's a toxin or nurturing behavior, all of these effects affect us in a very similar way at a molecular level by changing epigenetic variation. And so it'll be very interesting both to expand kind of the realm of different experiences that can induce these effects, to look at the heritability of these effects, but also to explore these in different species, including humans, and that's becoming increasingly possible. And one final point is that epigenetics raises the issue of plasticity versus stability. DNA methylation is very stable. It can maintain gene expression patterns across the lifespan, but it is potentially reversible. And if we can target that reversibility, it may be possible to kind of remove the layers of effects of early life adversity and set things anew. And so working with the National Institute on Aging, we're trying to look at how later life interventions can target these epigenetic pathways and lead to healthier aging outcomes. So I'll stop there, but just wanted to thank, of course, all my colleagues and students at Columbia University, my collaborators at the Columbia Centers for Environmental Health, and Catherine Monk and Ben Tycho, who we're working on the prenatal stress work with. And thank you.
One second there. Hi, I'm Kelly Lee from Ann Arbor, Michigan. I'm a private practice lactation consultant, so I'm kind of community-based right now, but moving towards research. And I was just wondering if you could talk a little bit about why you were focusing all of this on the licking grooming behavior rather than the nursing behavior, and if you can talk a little bit about why that was a more powerful indicator for tracking versus nursing.
Yeah, that's interesting. In some of the initial studies that we did, it was actually nursing that was a stronger predictor. But in the end, it was apparent that it was because nursing was so highly correlated with licking, we couldn't really disentangle them. Now, part of the answer is probably methodological, in that it is much easier. So, I tried to train the lab in trying to look at different nursing postures in our rodents and different frequency of nursing. But really, when we're watching nursing behavior, we're not looking at how much milk the offspring are getting. We're not even looking at milk letdowns. We're looking at, physical contact, which is going to be a collection of all of those events. And so probably it's a very gross measure of what's going on. So perhaps if we could target within the mother-infant interactions during nursing more precisely, it might be an interesting predictor of outcomes. But for the most part, when we look at licking and grooming versus nursing as a predictor, it's licking and grooming and not nursing that predicts most of these outcomes. And I think it's the distinct kind of tactile nature of the of the behavior that's quite critical for that. Whereas nursing, obviously very important for growth, but perhaps not for the specific outcomes that we're looking at in these studies.
It's interesting because it reminds me of, in humans, that you can have a mother who is breastfeeding but who is obviously not have this sort of intense bonding and attachment. They can be feeding the baby at the breast, or they might choose to pump and exclusively bottle feed their milk, but they don't have the same kind of tactile relationship with that child. So it's interesting to me that to say that it's more important, the touching is more important than it is the actual feeding.
Well, certainly, we haven't kind of done a duel between them, and I'm certain that there are elements of the nursing that are advantageous for certain outcomes. But I think kind of in the context of the work we do, and certainly in the species that we're working in, this is their primary form of tactile stimulation from the mothers. And, if you look in the human literature on depressed mothers, what you see invariably is that the way in which they touch their infants is different. And they reduce these. And, stress is a big inhibitor of tactile stimulation, not just between mothers and infants, but between any two people. And it probably relates to how stress and oxytocin work together. And, I think certainly the nursing and licking or the tactile stimulation will probably go hand in hand to some degree, because touch increases oxytocin, oxytocin increases lactation and nursing. So there's probably a feed-forward effect of tactile stimulation that enhances the nursing experience.
Yes, it's so terrific. I have acute rodent model envy now as somebody who works with human studies. Could you comment a bit on some of the issues you talked about working with Catherine Monk to move this into sort of the human area, on some of the challenges that you encounter now trying to go from animal model to human studies?
Right. So I'd say there's probably maybe three key challenges. So first is capturing the right developmental time point that maps from our rodent work to the human work. So, if you think, for example, of the postnatal mother-infant interaction work that we've been looking at, the brain of our rodent pups during the postnatal period is far more comparable to a prenatal time point in human brain development. And looking at mother-infant interactions at a prenatal period is actually very difficult. You're really looking at placental function and hormone transfer and these kind of things. So I think trying to get that right is very difficult. When it comes to the epigenetic pathways, there is increasing human studies trying to look at epigenetic effects of adversity. So there are some publications now showing, for example, that orphanage-reared compared to family-reared infants have different genome-wide methylation patterns. The more adverse your experiences are, the more DNA methylation you have. Now, these results come from analysis of blood samples. And as I mentioned, tissue specificity is a really important characteristic of epigenetic factors. And what we don't know is how well what we see in the blood predicts that in the brain. So it may be useful for certain purposes. It may not be useful for understanding some of the pathophysiologies and behavioral disorders that emerge as a function of early life adversity. And I guess related to that is the third issue, is accessing brain tissue. Now, there's been some human work on this, but it's reliant, of course, on postmortem brains, which has many, many caveats. And for the most part, are based on suicide, individuals that have committed suicide. But even there, if you look at the brain of individuals who committed suicide but had a history of abuse versus those who did not have a history of abuse, you will see epigenetic variation that is predicted by that early life adversity. So I think the overall challenge, other than the whole lifespan issue that is very difficult to tackle, is that right now we know so little about these mechanisms at a basic level in an animal model, in a rodent, in the lab. It's hard to know where it will go and how we will be able to apply this to kind of intervene in humans and better understand the pathways in humans that are critical for these effects.
I've been working mostly with new parents, and I'm now moving more into those who are going to be talking to new parents. But what I find really hopeful, maybe, is that in the modern world out here, there's the, you should breastfeed, you shouldn't judge me if I don't world. And that the high licking and grooming not being necessarily dependent maybe on the breastfeeding gives some hope for those who choose for whatever reason or who can't breastfeed, that they can still provide a really great enriched environment for their children. And that there seemed to be real hope for those that came from a low licking and grooming environment. So even if you had adversity, that you can make potential changes. I mean, did I assess that right?
Yeah, I think certainly. I think there are multiple ways through which, a healthy brain can be achieved. And tactile stimulation, stress in the environment, nursing practices, later life juvenile interactions, all of these will feed in to shape, the individual. And so it's hard to think of all of them at once. We tend to, in any kind of experimental design, focus on one. But the fact is, there are many routes to the same outcome. And I think that's certainly true in this work. And I think the reversibility issue is really important. It's, I think both from a psychological perspective and a mechanistic perspective, that these things are not set in stone. They are not ingrained in your genetics. And even if they were it wouldn't matter because we could epigenetically shut them off. So, I think it does give some notion of kind of overriding even kind of prenatal adversities that you might not have any control over.
Francis, there is, I forget who did the study, but it was a rat or a mouse model where they had introduced a toxin to the environment and later removed it, and over several generations the methylation still persisted, or the epigenetic effect.
That's right.
So some toxins may not be, I don't know, have permanent inheritance effects, right?
Yes, I think that's true, and that was a pesticide, but there was fairly high levels of a pesticide. But of course, it does give you pause to think about, the things that we experience don't just affect us, don't just affect the next generation, but can turn up many, many generations later. I think in those models, though, there's been no attempt at intervention, right? So could there be some way to remove these epigenetic memories from ancestors past? And I think that's where the work could go that would be very interesting. And certainly there are pharmacological approaches that we can use. So for example, exposure to high versus low licking grooming, its effects can be completely reversed by pharmacological targeting of those epigenetic pathways in adulthood. So that gives us pause to think about what other things could do that. And I'd be very interested in thinking about how social enrichment could come and reverse those same pathways.
Kind of a comment to add to that, Francis. Tell me if I'm wrong here, but in the 50s when they gave the drug DES to women to prevent miscarriages, and the effects had an intergenerational transmission down to granddaughters where they had enormously increased rates of ovarian, bladder, liver cancer from that drug that was caused initially tremendously significant birth defects in the children.
That's right, yes. So in fact, DES and bisphenol A have very similar properties. And when they were considering DES as a I believe it was taken for an anti-nausea, I can't remember exactly, but I think they considered BPA, bisphenol A, as an intervention, but thought it was too toxic to use. Now we just make plastics out of it and drink out of them. But and so within the DES literature, there's a lot of interest in the epigenetic inheritance of it. Now the problem always is that looking at a maternal transmission, it's very difficult to know whether something is being perpetuated through this kind of germline or through the context, because mothers provide the context of development. Separating the two other than by some of the embryo transfer studies that we're going to be starting to do, is almost impossible.
Francis, can I ask you one more question?
Yeah.
So I hope I can describe the really elegant nature of your work and don't butcher it here, but I believe you showed that among the virgin females, those that were high licking and grooming had higher estrogen alpha receptor expression, and that was related to their latency to show maternal behaviors.
That's right.
Did you actually follow through to look at the neurobiological substrates of that you've connected to the estrogen alpha receptor expression to? So was that mediated through oxytocin, the oxytocin system, or I think you've also connected that estrogen alpha receptor to the dopaminergic activity in the rats? Were either of those two systems or otherwise active in the virgin females?
Yeah, that's interesting. So, people tend to look at one pathway or another. And certainly, as a consequence of receiving higher low levels of maternal care, both these kind of hypothalamic oxytocin estrogen systems are affected, but also the reward pathways within the brain. So we wanted to know whether the changes in estrogen receptor alpha in the hypothalamus were needed to change the whole development of that reward circuit, the mesolimbic dopamine circuit. In fact, it didn't. So what that suggests is that maternal care is basically affecting both of them at the same time, rather than sequentially affecting one and then the other. We certainly think the oxytocin receptors are downstream of some of these changes in the steroid hormone receptors, but it seems that there's independent pathways that affect the way that dopamine circuit functions.
Thank you very much, Francis.
Thank you.